about information in biology, and we will look a lot at the information in biomolecules like DNA, proteins, RNA. And we'll look at some of the machines, the molecular machines that we've discovered, that science has uncovered. And we'll try to talk about, how likely is it that these things could form by chance, given any amount of time? I think you'll agree at the end that there's very little chance these can form on their own by natural processes, regardless of the amount of time provided. Okay, just a little bit about myself beforehand.
Can you have the mic in?
Okay, sure. Boy, that's okay. I'm sorry. I am.
It's not mine, it's recorded.
Got it. I forgot yesterday, and I apologize. All right. So this is just my family. You can see my wife there. My daughter and I. She invited me to a Valentine's Day dance. So that's what we were doing right before we went dancing, and then my son and my granddaughter, Catherine. And this is my dad and mom. My dad has passed away, but my mother's still alive. She's almost 88 and lives in Texas, in Fort Worth, Texas. And so these are the two places I've ever lived, Texas and North Carolina, in 5 different cities: Fort Worth, San Antonio, Austin, Greenville, North Carolina, and now Raleigh. So that's my history. I have the chemistry emphasis. I used to work at GlaxoSmithKline, now a consultant. This is the kind of work that I did. I did what's called force degradation chemistry, where we take a new drug and try to understand how it degrades and the different compounds it turns into for safety reasons, so that people don't get poisoned when these things form, and also for analytical reasons, so we could develop methods to detect these things that tell the quality of the product. So it has something to do with shelf life. So I just wanted you to get a sense of something I did. That's what my office used to look like. This was my lab. These are the guys that I used to work with for a good 20 years. My boss is in the red on the left. And this is like one of the last times we were together. We were all laid off in 2014 from Glaxo when they closed the R&D division at RTP. But anyway, so information in biology. So here's a couple of good contrasting statements that kind of show the two different approaches to this whole topic. So the origin of life research. There are people out there who believe that life emerged on this planet by a series of chemical accidents. And they are trying to figure out how that happened. Chemically, how could that have happened? So their task is to find an algorithm, which is just a procedure or a solution, a natural law that leads to the origin of information. They understand the biggest problem facing evolution is the generation of information, where there is no information before. How do you generate information? And William Dembski, who is a very smart person in the intelligent design community, has kind of summarized the whole thing and said the great myth of modern evolutionary biology is that information can be gotten on the cheap without recourse to intelligence. He did his dissertation work on this. He's written several books on it and produced some videos. He's a really bright fella. And so anyway, so origin of information in biology according to neo-Darwinism. Now, what is neo-Darwinism? It's taking the idea that animals develop through variation of some kind, and then that variation, which happens to be adaptive, is selected by nature or by natural selection, and so it's retained. So if you have a variation in your being and it helps you adapt, then that natural selection will select that, it will be inherited and passed on to the next generation. Darwin didn't know anything about genetics or heredity. It was before his time. And once they knew about genetics, then they tried to meld what Darwin had said with genetics, and that became neo-Darwinism. And so the new dogma is that information is added to the genome by random genetic mutations and natural selection. So if there's some mutation, some change in your genetic makeup that confers some kind of advantage to you, and you can pass that on to your offspring, natural selection will favor that, and that particular mutation will be retained in the population and will carry on. And presumably over eons of time, this process develops new organs, new tissues, new organisms, everything that we know in the biological world, as opposed to have arisen by this mechanism. So now what do evolutionary processes have to explain? They have to explain the origin of life, the Cambrian explosion in the fossil record, and we'll talk about that. The lack of transitional forms, right? What's the intermediate form, the missing link between organism A and B? You know, how can you tell one came from the other? The origin of information in macroevolution. Now macroevolution is just molecules to man evolution. It's simple life forms being transformed into complex life forms through this mutation selection process over long periods of time. How do you make what is the origin of irreducibly complex molecular machines? And we'll get into what does that mean. What is an irreducibly complex molecular machine? But there are these molecular machines, we'll look at a couple of them that are pretty fantastic. Convergent evolution, what that is that it turns out there are organisms that they don't believe that have similar traits, almost identical traits, but they don't believe that their last common ancestor had that trait. So what it means is that that trait was evolved independently in two different lines. So that's convergent evolution, and we'll talk about problems with that. And then information found in the epigenome. And the epigenome is now, they found that there are heritable changes that are made in organisms that have nothing to do with DNA. That they're not part of the genetic code, they're parts of other things, cell membrane, sugars, and other things that are heritable, but they're not a part of the genetic code. Okay, so let's see. So chance or design, let's get a sense of what is this information we're talking about. If you wanted to flip coins and had a quarter, heads and tails, let's say you flipped a fair coin 100 times and you wrote down what you got every time you did it, tails, heads, whatever, and just wrote it down. And this is the answer that you got. This is what you observed. And there was 100 coin tosses, and each outcome has a 1/2 probability, right? There's 50-50 chance each time you throw it that you're going to get a head or a tail. And so in this particular example, there are 49 heads, 51 tails, 50 alterations between heads and tails. And that's kind of what you would expect from just flipping a coin randomly and writing down the results, right? Do you believe this? Is it right? Is the outcome a product of chance or something else? Looks like a product of chance, doesn't it? Well, if you turn all the tails into zeros and the heads into ones, which is what would happen on a computer, right? Because everything's zeros and ones on a computer. Is there a pattern? Is there a pattern? Well, as it turns out, there is a pattern. If you know base 2, if you can count in base 2, it's actually counting in base 2. It's counting in base 2. Binary number systems, not random, it is a pattern. And binary numbers are independent of a random coin toss. So the probability of getting this particular arrangement is 2 to the -100th power, right? Because every time you flip it, you've got a 50-50 chance. And then the next time you flip it, it's a 50-50 chance. And when you multiply that out, it's like 1/2 to the 100th power. 1/2 times itself 100 times is what it is. That's the probability of this occurring by chance. Obviously, this couldn't have occurred by chance. The probability is almost impossible. So what looked like a random event that had nothing in it but just a random sequence turned out to be something that had very specific information in it and a very improbable event. And this is what is meant by information. Something that has a very low probability but also corresponds to an independent pattern. The low probability is being one chance in out of 10 to the minus 100th power. That's a very, very tiny chance. And then the pattern is the binary number system. That's information. It's complex because it's low in probability. It's specified because it corresponds to an independent pattern. So it's what William Dembski calls complex specified information, CSI. And he says it's the hallmark of design. If you can detect CSI in something, you've detected design. And we're going to embellish this, but this is the basic idea. Now, he invented something called the explanatory filter for understanding the cause of something. And he has 3 different possible causes for anything that you might observe. One is just pure chance, right? One is law. There was no other option. If I drop a ball, it's going to go down. Law dictates, the law of gravity dictates that it fall to the ground. So that's another option. Law could account for some phenomenon. Chance could account for it. Or design could account for it. Is there a way to actually differentiate between those 3 alternatives? And he says yes, and this is his filter that allows you to do that. So you have something that's happened, and so you ask, is there many possible outcomes? Could any number of things happen in this particular case? And if the answer is no, there was only one possible outcome, then it had to be law that dictated the outcome. And so that's your cause. There's some law that makes this happen. You're done. You've figured out the cause. But if there were many possible outcomes, but you still just saw one, okay, then let's go down and ask another question. Is it complex? And what we mean by this is it unlikely? Is it unlikely? Is it very improbable? If the answer is no, if it could have happened, it's not that hard for it to have happened by chance, 1 in 10 chance, 1 in 100 chance, okay, it was probably just chance that caused this to happen. Then you're done. Chance caused this to happen. It's just a random fluke event. But let's say that it is very complex. It is not likely that it could have occurred. Then you ask the question, is it specified? Does this outcome correspond to some independent pattern that we know about? Such as in the example of the coin toss, and those outcomes corresponding to the binary number system. If the answer is no, well then you still could have something that occurred by chance. But if the answer is yes, then you've detected something that was designed. And this is and he has a whole mathematics to this, but this is an overview of his process. This is the algorithm for detecting if something is designed or not in the different alternatives that are there. Does that feel comfortable? Are you comfortable with that? Does that make sense? Can't tell. Okay, a few of yours nod your heads. Okay, so here's a good example of this. Here we have Mount Rushmore, and we have the side of a mountain. Both of these are very complex arrangements of atoms and rocks, yes? Both are very unlikely to just occur, right? But the top one, the shapes that we see correspond to an independent pattern. There are these 4 presidents that we recognize. Oh, there's some information there. On the bottom, it's just jumbled stuff. And while, yes, it is very complex, and yes, it is very unlikely, nevertheless, chance does account for this. It wasn't designed, it just happened. Right? So there's the difference in what we're talking about here. Okay, so this guy named Claude Shannon, long ago, came up with this idea of how can you quantify mathematically information. And he came up with this equation, and this is the only equation we're going to have, it won't get any worse than this, that I equals minus the logarithm base 2 of the probability of the outcome of something. So I is the information in bits. Logarithm, if you understand what a logarithm is, minus log of the probability. And so what does this mean? The information content of an object or event is equal to the negative logarithm of its probability. Another way of writing a negative logarithm is 1 over the log logarithm of the probability, base 2. Now, whether you understand the logarithm or not, what you can probably understand is that if the probability decreases, then what you're going to end up with is a lot more information. That's what this equation said. The probability decreases, you end up with a lot more information. Okay? Now, he used this equation. William Dembski came along later on and said, so what's the greatest number of possible, the greatest amount of information the universe could ever generate randomly? What's the greatest amount it could ever generate? So it said, well, let's think of the fastest possible process that we can think about in nature. Let's think about how many particles there are in the universe, and let's think about how old the universe will be at heat death. Because the universe is supposedly winding down, is going to die eventually because of the second law of thermodynamics, and will die of heat. If the Lord doesn't come back, it will eventually just wear out and die of heat death. And that's actually trillions and trillions of years from now, if that were going to occur. Trillions of years. We're not talking billions, we're talking trillions, and even more than trillions. So he said there's 10 to the 80th particles in the universe, estimated. You can read that in literature, it's all over the place. Then he says that you can have 10 to the 45th events per second. I don't know of any physical process that's that fast, but that's extremely generous. I think that's actually maybe the fastest that anything could occur, according to the Planck time. And then you've got the number of seconds, 10 to the 25th seconds. That's trillions and trillions and trillions of years. So when you multiply that out, what you end up with is 500 bits of information are possible by all the matter in the universe doing some process as fast as anything possibly could for the entire length of the universe's age. That's the maximum amount of information that could ever be generated by a random process. And William Dembski calls this the universal complexity bound for CSI. So in other words, if you can find something in nature that requires more than 500 bits or contains more than 500 bits of information in it, it means the whole universe, over its entire duration, with all its particles going as fast as it can go, could have never generated that thing that has that much information, ever. Could never have done. So then the question is, are there any things in biology that have more information than this 500 bits, this universal complexity bound? And the answer is all over the place. Yes.
The acronym CSI, yeah.
Complex specified information. Complex is unlikely. Specified means it corresponds to an independent pattern. Information. And so we're going to see how this plays out. So now this we already just discussed, so I'm going to move on. Now he came up with, after he did his dissertation on this, after he had done all his studies, he came up with what he calls the law of conservation of information. And it says information can be transmitted or degraded, but cannot be created by chance and natural processes. And by here he means complex specified information. The information content of a closed system remains the same or decreases with time. So what is he saying? If the universe is a closed system, then whatever amount of information it ever had was there from the beginning and had to be relayed through natural processes to some other forms. It can be translated into other things, it can be relayed, but you're not going to get any new information, not from natural processes. In other words, since the universe is a closed system, the information in DNA must have been present at the beginning and somehow translated into base sequences in DNA. Somehow, in the patterns of the atoms at the Big Bang, if you believe in such a thing, I don't, but if you did, that pattern, that imprint, over billions of years had to have been transferred to the biomolecules here on this planet, is what he's saying. That's where that information would have had to be. It would have had to have been there from the beginning. Okay. And even if that were true, as a deist might believe, where did the information come to begin with? It's still got to account for where it came from. Now, information in DNA. This is a schematic of kind of the basic information processing system in every cell that exists, that we know about. It's not that bad. There's three things here that you need to look at. DNA, RNA, proteins. DNA is converted into RNA, RNA is converted into proteins. DNA contains information and is where it's stored. RNA transfers information between different things in the cell, and proteins do work, can do chemistry, and they add structure to the cell. DNA, RNA, proteins. So up at the top left, the DNA, it's got 4 letters. All DNA is made of just 4 letters. There's a 4-letter alphabet in DNA. A, G, C, T. That's all you need to know. A, C, G, T, that's it. So DNA for human beings, there's 3 billion letters in the DNA for human beings. A, C, G, T in any number of orders for 3 billion letters, A, C, G, T. That's all it is. That's it. That's all the DNA is. A, C, G, T.
They do have names, though, right?
Oh yeah, they do. But for our purposes, we don't need to say those things.
Adenine, thymine, cytosine, and guanine. Now, what is it all of this stuff called?
Yes, sir. So what happens? Inside a cell, in the nucleus, if you have a eukaryotic cell, not all cells have nuclei. Prokaryotic cells don't have nuclei, but eukaryotic cells do. Inside the nucleus, the DNA, some part of that DNA is targeted, and it unwinds, and a machine that's made out of proteins comes along and reads the DNA, and it manufactures a copy of some segment of the DNA, and that copy is called RNA, or messenger RNA. Why is it messenger RNA? Because that little piece of messenger RNA is going to go outside of the cell membrane into an organelle called the ribosome. And the ribosome is a machine, it's a factory. It takes in RNA, and it takes in amino acids, which are the building blocks of proteins, and it reads the RNA, and it translates the information in RNA into a sequence of amino acids to make a protein. And then the protein is folded up in a specific way that makes it able to do its function, whether it's chemical catalysis or structure or whatever it is. Well, as it turns out, if you come back from the proteins, this thing called RNA polymerase, that's a protein. And that's what it polymerizes monomers when it reads the DNA, and it polymerizes those monomers into RNA. And so you have this cyclical thing. The DNA is needed to make the proteins, but the proteins are needed to read the DNA and make the RNA. So what's the chicken and the egg question? Which came first? Well, this is actually a system that this is where the term irreducible complexity comes in. Because if you remove any component of this system no longer works. It's worthless. This has, everything that's in the system is essential to its function. Anything's removed, it's no longer valuable. And so you've got to think about evolution. Evolution only selects things that work and add benefit. If it doesn't work, it's not going to select it. So if this thing doesn't work, then it doesn't matter if it's almost there and you're missing the RNA, it's just going to say, that's just junk, forget it, we're not keeping it. So you have to make all of this at one time or it doesn't work. So now you can start to get a sense of, oh, you've got to put all those pieces randomly, you've got to form them all randomly from some source, and then you've got to put them together in the same place at the same time, and they all have to work and mesh with each other for the system to work. Start thinking about what's the probability of that. Just getting one of these molecules alone by itself at all is really hard, but think of trying to get all of them in at the same time, and at the same place, and working in the right way. No, it's not going to happen. So here's the structures of RNA and DNA. Now, what's an amino acid? So you can see that top structure. All amino acids look like that, and that little G is a group, and all amino acids, the only difference is that what G is. There's 20 amino acids. They all look like that. So down at the bottom, you see a protein. It's a polyamino acid. Many amino acids strung together. You put them together, you lose water, and you put another two together, you lose water, and you get this polymer called a protein, and that's all it is. It's polyamino acids. That's it. And so how does this work again? The genetic code. So you've got this DNA, ACGT, in whatever fashion you want. And in the process of transcription, which we just talked about, in the cell nucleus, that RNA polymerase comes along, reads it, generates a messenger RNA molecule, and now you've got the messenger RNA that's down there. The only difference between RNA and DNA is that T now becomes U. Just letting you know that. Now, there's something, and I'll show you what this means in a second, is it turns out when you go to the ribosome, the way the ribosome works, and this is where the factory is that makes the proteins, it reads the RNA and generates the protein, the polyamino acids. It reads the RNA 3 letters at a time. Each 3 letters corresponds to 1 amino acid. So now you've got a different kind of language. You go 3 letters, and it reads that and says, ah, that's thymine, and it pops it in. And then it comes along and it reads another 3, and it goes, oh, that's serine, and it pops it in. And so at one end, it's reading 3 letters at a time, and on the other end, it's taking amino acids and saying, oh, I need this one, putting it together, I need this one, I'm putting it together, I need this one, and it's translating the information in the RNA into the protein sequence at the bottom. It's actually translating. It's almost like English into Spanish or something. It's processing information. Incredible. Incredible. And so you get this idea of what's going on. Now what I want to show you now is just a quick little video of this process that maybe will help you better understand it.
In the years since Kepler's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism.
There's your RNA polymerase.
Transcription. A molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a 2-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins.
So this is your amino acids. That's your RNA. The amino acids are being joined together at the bottom according to the code at the top.
Specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling.
Okay, so that was just to give you a good sense of what's going on and what we're talking about. And I just made a mistake in terms of where I was. Let's see. So that gives you a good visual sense of what we're talking about. Now, what we haven't even touched on is that's just the system that makes all of that stuff. Now there's a whole other system that decides when a protein's needed and when the RNA polymerase is sent to a specific place in the DNA to manufacture a protein. So there's a whole regulatory system on top of this that's making the decisions, when do I need a protein? And then there's, as it said, there's a whole address system in the cell that says, oh, I need the protein and I need it over here. So it picks it up and it takes it to that place and deposits it, and then it gets incorporated into whatever's there as it's needed. So there's a complete feedback loop system in the cell saying when it needs something and to turn things on and to turn things off. So I mean, it's just, there's layers and layers and layers of information and control and processing. Pretty unbelievable. Now, so let's look at this for a moment. So if there are 20 amino acids found in proteins, imagine making a 150 amino acid protein, one amino acid at a time. This is a modest sized protein. This is not a real giant protein, by the way. Each amino acid must be selected by chance, so the probability of selecting each amino acid is 1 in 20, right? There's 20 different amino acids. So each time, if you're going to randomly pick an amino acid, you've got a 1 in 20 chance of getting it, right? So the probability of selecting 150 amino acids, amino acid protein by chance is 1 over 20 multiplied by itself 150 times, right? Makes sense, right? So how many bits of information does that contain? It contains 648 bits. Remember, we thought about the universal complexity bound that Dembski came up with. 500 bits of information could ever be generated by the universe in its entire history with all its particles going as fast as it could. So this has more information already than the entire universe could ever generate in its entire history. So this exceeds the universal complexity bound. So William Dembski would say, this definitely says this was designed, not an accident. Okay, the simplest cell we know about is called Mycoplasma genitalium. It has 482 proteins. We just talked about how to make one protein that had 150 amino acids. This is the simplest cell science knows of. It has 482 proteins just like it. 482. And it has a DNA that has 562,000 bases in it. Some believe that a cell might be able to function with 250 to 400 genes. There are hundreds of proteins in a simple bacteria translation system. That was the ribosome. That's the ribosome. It looked pretty simple, but it's actually hundreds of proteins and different kinds of RNA. So there's 20 more in transcription, 30 minimum for DNA replication. So the simplest life needs DNA and proteins, also needs proteins for energy production and processing. So for the simplest cell, all the proteins, DNA, and organelles must be present at the same time and at the same place and configured correctly in order for it to function and work. This is what science has to explain. If you want to believe in a naturalistic origin of life, you have to explain how you get all of that just by accident. Now, as first life forms weren't that complex. Well, we don't know of any other kind of life forms. How do you know? Well, it just couldn't have been. Well, how do you know? Because it evolved. Well, how do you know that? Well, because that's the only way it could have come about. Well, how do you know that? Well, because we can't let God in the door. Well, why not? Well, that's not what scientists do. Well, why not? So, origin of life. It has a lot of problems. It has problems at every level. We just talked about assuming that you had all the amino acids prepared, pure and ready to go. But in any natural scenario, when you try to make these things, you get muck. You don't just get the 20 nice amino acids that you want. You get a lot of other junk. So you've got competing side reactions that you don't want. So there's the building block problem. How do you get just specifically what you want? There's side reaction problems. There's problems with oxygen. They've shown in experiments that when you try to mimic what you think is the ancient Earth and try to do this synthesis of these monomers, if there's any oxygen present, it shuts it down, doesn't work. But the other problem is, if you don't have oxygen, you don't have ozone. If you don't have ozone, UV light comes through, and UV light destroys the molecules. Oh, it's rough. So then there's the dilution problem. Put this in the ocean, you've got to make polymers. You've got to put monomers together. If there's one per cubic mile, they're not going to find each other, and you're not going to get you're not going to get a polymer. Chirality problem, we won't talk about that. Hydrolysis problem. This is once it's put together, water wants to cleave it back to where it came from. And if you're in the ocean, you've got lots of water. Sequence problem, this is what we've been addressing. It's the order, the sequence of the amino acids or the base in DNA have to be specifically correct in order to get a functional thing. Not just any sequence will do. That's the time problem. You know, we talked about the entire trillions of years. We're not talking about 4.5 billion years, that's how old they say the Earth is. We're talking about trillions of years, trillions of years. So here's someone who is actually an expert on the information in biology. He is not a Christian, but he is sympathetic to intelligent design people. He says the reason that a scientific explanation for the origin of life has not been found may be that the problem is beyond human reasoning powers. Life is consistent with the laws of physics and chemistry, but not derivable from them. We must take life as an axiom. The time of the molecular biologist is better spent in understanding life as it is, having accepted the inexplicable axioms of these subjects. Cambrian explosion. What is that? Well, that is a period in the fossil record that supposedly occurred about 550 million years ago, that all of a sudden, all these life forms start appearing in the fossil record, just out of nowhere. They seem to just come from nowhere. There's very few things living before them. There are a few, but very few. And then all of a sudden, everybody know what a phylum is? Maybe not? It's the body plans of organisms. When scientists classify living things, they classify animals by body plans. How's it laid out? Where's the legs and the head and the tail and the various things? And so there's groups of body plans. And we're in the chordata phylum, which means it's something that has a backbone. And so each thing that's in a phylum is very different from all the other phyla. Phyla are very distinct and different from one another. And it turns out that in the Cambrian explosion, 20 of the 26 known phyla for animals are there. And it's in what they call a geologic blink of an eye. Maybe in just a few tens of millions of years, compared to 550 million years, all those 20 phyla appear out of nowhere, and at the bottom of the fossil record, at the bottom. Now, if you think about that, it's a real problem for Darwin, because phyla are the greatest differences between organisms. Now think about Darwin's tree of life, right? You evolve a cell somehow through some chemical accident, you finally get a cell, and it starts to divide, and it starts to change, it starts to differentiate. And maybe eventually you get two different kinds of cells and two different species. And then after a much longer period of time, you get more variety of different kinds of cells. And maybe someday, eventually, you get multicellular organisms. And then you start getting plants and animals and amphibians and mammals and reptiles and all the things that are known to life. Well, so at the base of the tree, at the base of the fossil record, we should see simple, very few different types of body plans, right? Because you've only got the cell down there, that's it. So there should just be this simple thing at the bottom. And as you go up the fossil record towards present day, you should start seeing more different kinds of phyla and more different kinds of organisms until finally that tree starts off with a trunk and then it just branches and it just goes up, right? That's Darwin's tree, right? That's what you would expect. That's logical, right? That's not what's found in the fossil record. These are all the critters, drawing of the critters that they have at the Cambrian explosion. Now here's what I was talking about. On the left is what you'd expect from Darwin's theory. That if you look at the top, you'll see that you start off with very few phyla, and over time you build up more and more and more. And at the bottom you see that tree. You start with one phylum, and as time goes on going up, you get more phyla and more differentiated body plans, right? What's the actual thing that we do see? We see all the phyla at the bottom at the beginning. Boom, it's all there to start with. Not at the top where it ought to be, it's at the bottom. How can that be? How can that be? It's not supposed to be like that. So we've called this the inverted cone of diversity. It's upside down. It's the opposite of what Darwin would have predicted. The opposite. All that information, all that differentiation, all that evolution, it's already there at the very beginning. And the organisms are already very complex. And they have very complex eyes and organs and all kinds of things. So it looks more like a lawn than it does a tree. And as it turns out, since the Cambrian, we've had no new phyla emerge, and we've actually had some go extinct. We have less phyla now alive than they had back then. Just the opposite of what it should have been.
Then if I might insert some take-home messages, this is where, if nothing else, academic freedom should be allowed. You see, that's just information and academic freedom. But we allow the powers to be to say, oh, if you talk about religion, you can't say that. Gotta see. And there's where we, who are, whether we're atheists or religious or faith persons or not, we got to stand up for academic freedom there, at least. So put that on YouTube.
What? I think it's on YouTube. Good point. So let's see. Just trying to. So here's some of these fossils that they find down. There's some in China and also up in Canada, where they have found these fossils down at the base of the fossil record in the Cambrian. And they're typically very odd creatures, at least from what we would say and what we've seen. But they're very complex. They're already well differentiated. The trilobites have compound eyes like insects, and they have 360-degree vision. They've been able to determine that. This is at the base of the fossil record. Where are the precursors? Where's the evidence for evolution? How did we get all this differentiation at the beginning? So there's the Cambrian. So this is the fossil record, and that little black space at the bottom, that's where all that happened. That little tiny little piece of time, everything. So, so the cause of the Cambrian explosion must explain how much new functional DNA, right? You've got new functional DNA. When you come up with a new organism, you've got to have a new code in the DNA that codes for all the different organs and the different things that's specific to that organism. So that original organism has got to go through transformations of infusing new information and generating new information at the molecular level in order to create all these new organisms that create all these new phyla. So you've got enormous amounts of new information that have to be generated in a short period of time at the base of the fossil record. So there's new functional proteins, new cell types, new tissue types, new organs, new body plans. And so you've got to do all that quickly without leaving any evidence of transitional forms or precursors, because there aren't any of those. So there aren't any intermediate forms, there's no precursors, and it all happens at the beginning, but we're told that that has to have been by natural process. The random mutation natural selection mechanism can't produce a few functional proteins in billions of years. It is also unable to account for all the new information formed during the Cambrian explosion. However, a designer could generate that much information. We already know that intelligent agents can generate complex specified information from observation. We see people do it all the time. That's empirical. That's not religious. That's just common sense, right? We see people do that. So why can't you apply that same reasoning to this? They don't have any answers to this. They don't have experiments that they've done in a laboratory where they can say, oh, we've done this in the lab, here's the answer. They're not even close to that. They're still talking about how do we get ACGT naturally without it being destroyed and without it getting a bunch of other junk in the test tube at the same time. Let alone how do you put all those things in the right sequence and put them all together at the same time. Don't have any idea about that.
Another just to take on just a quick, when I started teaching biology 50 years ago, we were going to talk about the simple and complex. But with the DNA knowledge and the complexity of DNA that we found in the late 60s, we understood then it was complex. And yet the evolutionists had already gotten a hold, you see, with simple fossils, complex fossils, or an amoeba to a man type model.
So, yeah, just to give you a sense of, even it gets worse. It gets worse. Because as it turns out, there's multiple messages on one strand of DNA. We talked about reading the DNA and making the messenger RNA and then becoming a protein. Well, it turns out that in that one strand of DNA, there's not just one protein coded there. There's several proteins coded there. You can read a shorter transcript. You can read a longer transcript. You can read different transcripts from different parts in the genome, and then the machines will bring the different parts together and stitch them together into a new RNA. And then that will go to the ribosome and make a protein. So it's not just a linear reading. It can actually sometimes be read backwards. The back side has got the information. The front side's got the information. Chop it up several ways, it's got the information. It recombines it different ways to get new information. All that's programmed in there and already there. So the orders of information, it just explode. Let's see. So here's the idea. So up at the top you see position number. This would be like in a DNA strand that's 100 units long. And so these are the different possible codes that come out of that one strand of DNA. You've got multiple proteins that can come out of there that are called upon to be generated and manufactured by this regulatory system when they're needed with these feedback loops. Oh, we need this one, we need that one, go make it here, go make it there. Read there, then stop, read there, then stop. Read all the way over here, then stop. Make it over here, make it over there, bring these two together, join them together, now send that to the ribosome. Now take it over here, but next time take it over there. It's mind-boggling. So this is the idea of you're reading this transcript at the top, and then you get the original translated version, the transcribed version, and then you're editing it afterwards. And you're taking out the checkered parts, and then recombining the black parts to make the final transcript that goes to the ribosome. So it's just giving you an idea, and I'm gonna end with this. This will be the last thing I said.
Fine.
And this is a good lesson in how dogmatic science can get it wrong. And what evolutionists have said for a long time, that in our DNA code, there's a lot of junk that's left over from eons of evolutionary dead ends. They said, well, only 5% of the DNA is actually ever used to make proteins, which is true. The code in only 5% of our DNA to make to make proteins, that is correct. And they just assume, well, the rest of it doesn't have any function, it doesn't work, it's just junk, it's left over. It's exactly what we would expect if an evolutionary process had occurred. Why would a creator put 95% junk in a genome? Doesn't make any sense. And not only that, but since we believe in descendant modification from a common ancestor, that all human beings came from some ape-like hominid in the past, and we compare the DNA sequences of the chimpanzee with the human being, and they look so similar, and both of them are full of junk, how could it be that God would put junk, just the same type of junk, in both of these organisms and then not be related to each other in an evolutionary way? How could that be? Doesn't make sense. Evolution's proven. Creation's washed up, it's gone. So this is Richard Dawkins. Genomes are littered with non-functional pseudogenes, faulty duplicates of functional genes that do nothing, while their functional cousins get on with their business in a different part of the same genome. And there's lots more DNA that doesn't even deserve the name pseudogene. It too is derived by duplication, but not duplication of functional genes. It consists of multiple copies of junk, tandem repeats, and other nonsense which may be useful for forensic detectives, but which doesn't seem to be used in the body itself. Once again, creationists might spend some earnest time speculating on why the Creator should bother to litter genomes with untranslated pseudogenes and junk tandem repeats. But then in 2012, something called the ENCODE Project published a whole bunch of papers in Nature, which is one of the best science magazines in the world. And they came out and they had done a survey of the non-functional, the non-translated, the non-coding DNA in the genome. The part that doesn't get translated into proteins. And they found that 80% plus most likely has a function. And since that time, they've been finding those functions and what they are. And as it's looking like now, the trajectory is that almost all of it's going to have a function. And so we talked about the regulatory superstructure, what turns on when you make a protein and where you take it and all those things. That's what all the rest of that DNA is doing. It's the control mechanism for the generation of the rest of it. Okay, so I'm going to leave it there, and thank you for your attention.